Okay, so I really like the way this program has been set up because we have a presentation that gives us the real-world problems and the issues that communities and leaders are trying to struggle with to meet water sustainability, and then we've got that balanced with a science presentation. I like this format with the science translation as we talked about this morning. And our next presenter is Pedro Alvarez. You probably all know Pedro. He received the Clarke Prize in 2012. He's been a leader in water resource sustainability and water pollution control for quite a long time. He's been focusing on both bioremediation and environmental nanotechnology. He did get his degrees in Michigan, I saw. But he's been at Rice University for 2004, since 2004, where he runs several centers. He's going to be talking to us about antibiotic resistance, which I think is one of those emerging contaminants or emerging concerns that we don't quite know what to do about yet. Pedro, please.
Thank you. And how do I get these slides to come up? What?
Did you have a link in your presentation?
No.
Okay.
No, it's okay. Anyways, good afternoon. It's a great pleasure to be here today and celebrate David's honor and see so many friends and opportunities for networking. This is 2014. It marks the 100-year anniversary of the activated sludge. I don't know if you knew that, but about 100 years ago, Arden and Lockett invented this wastewater treatment process and demonstrated it in Manchester, in England. And over these 100 years, it has done a lot of good for water pollution control and for preventing the spread of disease. However, things are changing, and not only the activated sludge, but also in general, the water and wastewater infrastructure are facing many new challenges. One of those is, of course, contaminants or emerging concerns, and I think it's appropriate to think about antibiotic resistance genes as an emerging contaminant. And, so what I want to do today is basically show you some evidence that, unfortunately, some wastewater treatment plants are serving as breeding grounds and point sources of discharge for these antibiotic resistance bacteria, and then offer perhaps some potential solutions to help us mitigate with this problem. Let me begin with a little bit of background of why this is important. As you know, microbial control is a growing concern. It's a great opportunity to protect public health that is becoming more and more important as we consider the growing need for water reuse. But many places in the world are not doing a very good job at that. 1/2 of all hospital beds in the world are right now being occupied by someone who got sick by a waterborne infectious disease. And in the US, this is gaining renewed relevance, due to the emergence of these antibiotic resistance bacteria, which are a result of the abuse and misuse of antibiotics. But the point is to put the magnitude of this problem in perspective. These infections that are resistant to antibiotics are costing us every year $20 billion in the US. They represent 8 million hospital days extra, and they cause the death of 23,000 Americans every year. So it is an important problem, and guess what? We environmental engineers can do a lot about it. So, the mechanisms by which bacteria defend against antibiotics are actually pretty well known. I mean, this slide that I'm showing you is from 1998, right? And one of the common approaches that they have is that they create these systems to spit out antibiotics or heavy metals that harm them, the so-called efflux pumps. Other bacteria, they produce proteins that protect and shield the ribosome, which is a common target by some antibiotics like tetracycline. But the one mechanism that is quite challenging to address, that the one I'm talking about, is the production of some enzymes that degrade the antibiotics. As an example is this enzyme called the New Delhi metallobeta-lactamase. which is coded by the NDM1 gene. That enzyme can hydrolyze these beta-lactam antibiotics and render them ineffective. By the way, these beta-lactam antibiotics are the last line of defense that we have right now against infectious diseases. So this is a concern. Of course, by the way, the reason why this is called New Delhi beta-lactamase is because this was isolated from a multidrug-resistant bacteria, a Klebsiella pneumonia, that was isolated from a Swedish patient in New Delhi. These countries that have relaxed prescription policies for antibiotics and where there is a lot of medical tourism, where perhaps plastic surgery is cheaper and people go there for those purposes, then that contributes to the spread of antibiotics. And of course, the problem is that these antibiotic resistance genes jump from one bacteria to another, and that contributes to their spread. And this is becoming an international issue because they have been detected pretty much in every continent, perhaps except Antarctica. And, but this is, again, a growing concern. So we ask ourselves, well, are these multi-drug-resistant bacteria reaching wastewater treatment plants? And if so, what is happening to them? Are the wastewater treatment plants removing them? And if so, in which process in the treatment train are they being removed? Or if not, are they growing? Are they proliferating? And what happens to these genes if they are discharged to the environment? So that's kind of the questions that I'm heading to answer today. And first of all, I decided to study this problem in China for a couple of reasons. One is China happens to be the major manufacturer and user of antibiotics in the world, so the chances of these antibiotic resistance bacteria reaching wastewater treatment plants are higher. But the second reason is actually funding. I couldn't really get funding here in the US to do that. I asked the EPA and they said, no, go to USDA, and USDA said, no, go to NIH, and so on. So I said, I didn't try NSF, I should have. But anyways, the point is that we went to China. And the area that we studied is near Nankai University in Tianjin. Tianjin is about a couple of hours east driving from Beijing. It's the main maritime area. There is a river here, the Bohai River, the Haihe River that discharges to the Bohai Sea, which is the main port that Beijing uses. And you can see here the wastewater treatment plants in red, the ones that we sampled. This is a general reminder of how the activated sludge process works, along with a hypothesis. The first thing you encounter in wastewater treatment plants is a primary clarifier after the racks remove the floating objects. And this is, the purpose is to remove suspended solids. And then, of course, if you have antibiotics and antibiotic-resistant bacteria, some of them are going to break through and enter the aeration basin, which is where biodegradation occurs. And here, bacteria are going to grow and proliferate, and perhaps some of the antibiotics will be removed by biodegradation or adsorption into the biosolids. Then the secondary clarifier, the purpose here is to separate the biosolids and clarify the effluent, and many of these bacteria are recycled back to the aeration unit, and some are wasted. So we would expect that if bacteria are growing, the sedimentation is going to concentrate them, and you're going to have a lot of antibiotics-resistant genes present in the biosolids. And then you disinfect. In China, that's commonly done by chlorination. And of course, then you would have the effluent. And the question is, are there these antibiotic resistance bacteria still present in the effluent? And if so, what happens to those genes after the bacteria die? So we got a lot of help from Nankai University students, and they sampled the whole treatment train, the entrance, the inlet, and the outlet to every single unit. They have more units there, and these are our results. So we were measuring the total flow, trying to do mass balances also on the genes. And what we found essentially is that for every antibiotic resistance gene that was coming into this plant, 4 to 5 were coming out. The plant was a breeding ground for these antibiotic resistance genes. They were coming out primarily in the water sludge, in the biosolids that are applied to agricultural fields. And, but also, as a percentage of the total heterotrophic population, those that were resistant to antibiotic were much higher in the effluent than in the influent. One thing that worried me was that when we looked at what happens after chlorination, we noticed that these multidrug-resistant bacteria, some people call them superbugs, were also resistant, much more resistant to chlorination than the other bacteria. And as Urs told us this morning, it has nothing to do with them sharing the same resistance mechanism for antibiotics as it is for chlorine. It has to do that they are coincidentally more resistant to oxidative stress. And this is a concern. Now, what happens to them? Well, right around that time, with my colleagues at Nankai University, we were developing this new method using an internal standard. It's a gene from Arabidopsis, that is involved with the synthesis of cellulose. But anyways, this allowed us to differentiate internal from external DNA. By that I mean DNA, or antibiotic-resistant genes, that are inside a bacterium that is alive, versus the gene that has been released after the bacteria dies, all right? And what we're seeing here is two things. If we look at the colors here, the green and the yellow, is that the bacteria are predominantly present, not in the, this is, by the way, data from the Haihe River, the receiving waters of the effluent. So the genes are mainly present in the sediments. Of course, bacteria settle, so they're going to go to the sediments. But they die, and the external DNA, the antibiotic resistance genes that have been released after the bacteria die, persist for quite a long time. And that's because the clay in sediments stabilize DNA. So the question is, that those genes that are there, that are outliving the bacteria, can they be uptaken by indigenous organisms? And do they become resistant? The answer is yes. We are showing here an example using a kanamycin antibiotic resistance gene that we introduce artificially using a plasmid, and we can see that the gene is capable of transforming indigenous bacteria. So that's a concern, because it means that these bacteria that are being discharged can propagate resistance to indigenous bacteria. As an aside here, I work a lot in my lab studying how engineered nanomaterials interact with bacteria, and what do they do to activate sludge or wastewater treatment systems. And one thing that we notice here is that there are some nanomaterials that, at sublethal concentrations can damage bacteria and enhance the permeabilization of them. They make them more permeable and therefore more susceptible for the uptake of antibiotic resistance genes. So the point here is that there may be other things in wastewater, for example, polyethylenamide or something like that, or other chemicals that may enhance the permeability of bacteria, the permeability of the cell wall and cell membrane, and facilitate the transfer of antibiotic resistance genes. So anyways, given this background, then the question is, well, what can we do? We're engineers. We're supposed to not only describe problems, but also solve them. What can we do to mitigate the propagation of these bacteria? So the first thing to remember is that bacteria do not carry excess luggage. We have to remember that these antibiotic resistance genes are often present in mobile elements, plasmids, integrons, and so on. And there is a cost for maintaining these genes and reproducing them, an energy cost. So if they don't need them, they'll drop them. Okay, so let's remember that. So as an example here, working with tetracycline resistance, which is safer for students to work with than NDM1, we can see in these column experiments where we're trying to emulate what happens to the discharge from an animal farm, we see that when we remove tetracycline from the influent of the columns, resistance is lost. And there are other studies, for example, in Finland, where people rotate antibiotics for the purpose of maintaining the bacteria off guard and staying ahead of them, and that results also in the decreasing antibiotic resistance. So we also obtained some interesting data recently where we are looking at a correlation between the amounts of antibiotics that are present in the wastewater treatment plant and the relative abundance of antibiotic resistance genes. So the y-axis here is not the concentration of antibiotic resistance genes, but the relative abundance, normalized to the total bacteria. And as you can see, they are increasing with the, by the way, the white lines here is samples taken in the summer, the black symbols are in the winter. So what we are seeing here is a correlation that suggests that residual antibiotics are exerting selective pressure for the antibiotic resistance bacteria, which suggests that we need to try to either remove them, the antibiotics, before they enter the plant or use less. But I do recognize that correlation does not prove causation, and that there may be other factors that may also fortuitously select for antibiotic resistance, for example, heavy metals. Nonetheless, this suggests the need to minimize antibiotic misuse, abuse, and discharge which is very, very important. Now, also, in the US, let's remember that 70% of the total antibiotic use here is for animal agriculture, to make animals grow faster and get fatter. So that's another potential opportunity. You well know that technology is not going to be able to solve all of the problems. A lot of times policy is far more effective, and this is one area that I think we need to look into. Another important aspect is to recognize that under anaerobic conditions, which are thermodynamically least favorable for microbes to harvest energy, that can increase the metabolic burden of bacteria to maintain and carry that plasmid. So showing here this example using a facultative bacterium, E. coli, we see that this bacterium loses the antibiotic resistance faster when we subject it to anaerobic conditions. So this suggests that in these animal farm applications, maybe passing the drainage through an anaerobic lagoon might be a good idea. Or maybe in wastewater treatment, maybe a longer residence time through the anaerobic digesters might be a good idea. Along this line, if you make a bacteria grow faster, for example, by increasing the dilution rate in a chemostat, they also begin to lose the antibiotic resistance faster. Why? Again, consistent with the hypothesis. You increase the metabolic burden, it takes more energy to grow faster, then they drop the plasmid. And also, how much food the bacteria get affects their ability to harvest energy. If you grow them in a nutrient-rich, medium with a lot of food, they maintain the plasmid longer than if you subject them to starving conditions under a minimal basal media. So this suggests, again, that in the activated sludge, maybe adjusting the food to microorganisms ratio might be something that we should look at as a potential strategy to minimize the spread of resistance. So overall, we can see that there might be several operation variables that we may want to play with to mitigate resistance. For example, again, either decreasing the food to microorganisms ratio or the residence time in anaerobic systems and so on to increase metabolic burden. Of course, the other angle here or side of the equation is make our barriers, our our disinfection practices more effective. As I showed you earlier, chlorination wasn't doing a very good job, not to mention the collateral damage of disinfection byproducts that might be harmful. And as an example here, there are many advanced oxidation processes that could do the job. Urs also mentioned several, but I'm going to show you one that I have worked with, which relates to the use of fullerenes that have been modified. These fullerenes have the advantage of being excited by sunlight to produce reactive oxygen species that are very capable of disinfecting. And in this particular example, we add a positive charge to attract the negatively charged bacteria and the negatively charged virus. Yeah, I'm almost done. To the war zone. In other words, use a bait and hook approach to enhance the efficacy, and you can immobilize them with a covalent bond here to reuse this catalyst, and that lowers the cost and minimizes exposure and leaching potential. So the one collateral benefit is that some of these advanced oxidation processes, in this case nanophotocatalysis, can also destroy contaminants of emerging concerns like endocrine disruptors that are currently breaking through the plants. And it's very easy in the US to retrofit our current wastewater treatment processes because they already have UV disinfection often, and it's just a matter of converting that little reactor into a fluidized bed with like beads that have this photocatalyst attached to polish the effluent and mitigate this problem. That's very easy to do. We developed, for example, this 5-gallon reactor that is something that you can put on a plane. This is some work that we were doing for FAO, the Food and Agricultural Administration in Swaziland. And with a contact time of 30 seconds, we were able to provide water for peri-urban irrigation for 15 households. And not only was this removing 99% of the bacteria, but also removing a very high percentage of pesticides that were also present in the water. So anyways, to wrap it up, I hope I have shown you that some wastewater treatment plants can serve as breeding grounds and point sources for these multi-drug-resistant bacteria, and current disinfection processes by chlorination have a lot of room for improvement. So it will be up to us to contribute to technological development of greener and more effective disinfection approaches, and as well as to advance our fundamental understanding of the factors that affect plasmid and antibiotic resistance maintenance. For example, how to manipulate operation variables so as to increase the metabolic burden and force bacteria to drop the antibiotic resistance. With that, I want to thank my main collaborator here, who I misspell, I apologize, it's Luo Yi, not Yo Lo, and also the students that contributed to this effort, and I also want to thank you for your attention.
Testing? Yeah, there we go. Okay. So, very interesting, Pedro. Have you thought of solutions, a membrane bioreactor, where you'd have greater filtration combined with a potentially very long sludge age, and composting of solids as also important considerations?
I think that certainly will be a very fertile avenue to pursue now that you mention it. I have not done it yet because, as I told you, we just began to scratch the surface here, but I think that holds a lot of potential. Thank you for that comment.
Yep.
Very interesting talk. I appreciate it. Do you have any sense of the relative contribution of institutions like hospitals, to particularly like the beta-lactam resistance genes, as opposed to out in the general population? And if hospitals are in fact a significant source, are there control measures that could be pushed upstream?
Excellent question. Yes, there are a lot of studies separately that have indicated that that's a significant source of generation and propagation of antibiotic resistance. And indeed, I mean, this is consistent with the earlier talks that we had for the need for considering more decentralized approaches and not only like intercept the antibiotic resistant bacteria before they leave the hospital, but also the residual antibiotics that are excreted. So I think that that's an excellent approach, like a pretreatment like we do for industrial waste. Right.
One more question over here.
Thank you, Pedro. What about animal feed operations? How, especially in the U.S., they use, especially for cattle, they use a lot of antibiotics in their feed. How does that? What's the impact of those waste lagoons?
Yeah, that's a huge factor for the resistance of antibiotic genes. They are often, I have not seen much work pointing to multidrug-resistant genes, but the typical tetracycline resistance and so on, those are quite common there. And again, that's for many years, many people have worked in trying to change that and trying to use both carrots and sticks to minimize the use of antibiotics by the industry.
So in the last part of your talk, when you mentioned about the photocatalyst, I guess those hydroxyl radical single oxygen are generated on the surface of the catalyst. So what would be the impact of the attachment efficiency of this antibiotic? bacteria or their impact on the treatment efficiency?
So, surprisingly, when you attach the photocatalyst to a surface, the activity actually increases because it prevents agglomeration and it prevents just clumping. So you have a more surface area of photocatalyst available for the reaction. But it is also quite possible, as you point out, that there's things that may coat them over time, humic acids, antibiotics, or whatever. So that points to the need to regenerating them over time, but of course we're only beginning to do that. But there are other approaches, I'm sure, like ozone maybe, if you don't have bromide, of course, or you don't make bromate. But there may be a ferrate, there may be other advanced oxidation processes that could be considered depending on the economics. The photocatalyst I like because it can be accessible in areas where there is no electricity, at least the ones I'm using, which is usually when you need disinfection the most.
Okay.
Thank you.
Very much.